Thank you for joining today's ASBMR member spotlight session. This series was implemented by the ASBMR Diversity, Equity, and Inclusion Committee. This series aims to highlight the research of ASBMR members in a collaborative setting and aims in particular to shine a light on the research being done by ASBMR members belonging to groups historically underrepresented in biomedical research. Today's meeting will spotlight two ASBMR members. The first presenter will be Dr. Joy Wu, Associate Professor and Vice Chair of Basic Science at Stanford University School of Medicine, and she will be presenting her research focusing on stem cells as a source of osteoblasts for skeletal regeneration. Our second presenter will be Dr. Saravana Ramasamy, the group head at Imperial College London, and he will be presenting on the topic of functions of blood vessels in the skeletal system. Each presentation will take approximately 20 to 25 minutes each, and following each presentation, there will be an opportunity for Q&A and discussion with the audience. As a reminder, please keep yourself muted throughout the meeting. You are welcome to unmute yourself for questions or comments following each presentation. You may also submit your questions in writing via the chat feature. I'd now like to invite Joy to introduce herself and present her and begin her presentation.
Thank you, Hannah, for that introduction. And I want to thank the ASBMR DEI committee for this opportunity. It's always a great pleasure to share our work and nice to see so many friendly names among the Zoom participants. And I'm also very excited to hear about the Ramasamy lab research after this. All right, Hannah, can you confirm my slides look okay?
Yes, slides look good,
right.
Joy.
Terrific. So today I'm looking forward to talking to you about some of our work on stem cell sources of osteoblasts. A basic premise driving the work in our lab for many years has been that if we had a source of unlimited osteoblasts, we could do all sorts of very cool things. We could study disease mechanisms in vitro. We could do high throughput screening, for instance, for novel therapeutic compounds. We could develop cell-based regenerative therapies for conditions such as fracture non-union osteonecrosis, traumatic orthopedic injuries, or tumor-mediated bone destruction. And we and many others have been working on various stem cell sources of osteoblasts. And today I want to touch briefly on the four different approaches that we have been using in the lab, studying sort of endogenous bone marrow mesenchymal stem cells, of course, pluripotent stem cells, either embryonic or induced pluripotent stem cells, and finally some more recent work that we have done on direct reprogramming. So first, to just quickly summarize some of our work on MSc FATE commitment. This is mostly published, so I'll go through it quickly. As you all know, MSCs give rise to several mesenchymal lineages, including, of course, osteoblasts, but also adipocytes, chondrocytes, and other cells. In our lab, we have been very interested in PTH receptor signaling. As you're familiar, PTH receptor is a G protein coupled receptor. Ligands are parathyroid hormone and PTH-related peptide. As a G protein coupled receptor, PTH receptor talks to several G proteins. We've been particularly interested in the G stimulatory protein. And we have in the lab been studying mice that are lacking either the PTH receptor or the alpha subunit of the G stimulatory protein, GS-alpha. In early osteoprogenitors marked by expression of ostrix. We have published that these mice that lack PTH receptor signaling in osteoprogenitors develop pretty severe osteoporosis. As an example, here are mice with conditional deletion of GS-alpha in ostrix expressing osteoprogenitors. They have fractures of long bones and the rib. Here's a skeletal preparation at 1 day of life of a control littermate. And it's knockout littermate, and I think you can see that every rib has been fractured by the process of delivery. We've gone on to show that this is a dramatic reduction of both trabecular bone, as you can see here at day 7 in the proximal tibia. Control bone has abundant wild type, abundant spicules of trabecular bone that is nearly absent in the knockout. We went on to show that this was a failure of bone formation rather than excessive resorption, with a persistence of abnormal woven bone. In these mice, we also find a dramatic accumulation of marrow adipocytes. Here is the secondary ossification center of the tibia at 2 weeks of age in a knockout. And if we quantitate either the number of adipocytes or the percentage of fat by DEXA scanning, I think you can see that there's a significant increase in the conditional knockouts. And in mice in whom we use the doxycycline to delay pre-mediated deletion until after birth, at 6 months of age, you can see there's a very substantial increase in marrow adipocytes. Just to summarize our working model, we have found that these mice have an increase in sclerostin expression. That's not surprising since PTH is a known negative regulator of sclerostin. And we've been working on the model that this sclerostin affects Wnt signaling, which has been implicated in the commitment of mesenchymal stem cells to osteoblast versus adipocyte lineages. But mostly today, I want to focus on some of our stem cell-based works with pluripotent stem cells. And so when I was finishing up my postdoctoral fellowship in 2006, Yamanaka published his striking paper that induced pluripotent stem cells could be derived from somatic cells simply by the expression of 4 factors. And so we became very interested in whether these induced pluripotent stem cells could be a source of osteoblasts. And at the same time, we derived embryonic stem cells from mice as well. Many groups have been interested in differentiating pluripotent stem cells into the osteoblast lineage. And I want to highlight this important paper from Pam Roby's group in 2014. They had been working on human-induced pluripotent stem cells and differentiating them towards osteoblast lineage under a variety of conditions and using different cell lines. And what they showed was that in vitro assays of osteoblast differentiation, for example, expression of markers such as RUNX2, alkaline phosphatase, type 1 collagen, in vitro mineralization, that they could get reliably expression of RUNX2 and type 1 collagen. These are in both 2 lines of human iPS cells and bone marrow stromal cells as a control. And you can see that RUNX2 and collagen 1 expression are pretty reliably induced. However, expression of other markers does not seem to be as reliably induced in iPS cells. But you can find evidence of in vitro mineralization, and this is often used as an assay of osteoblast function. However, the important assay that they did was to then transplant these so-called osteoblasts into in vivo to see if they could form bone. And you can see that while bone marrow stromal cells give rise to in vivo bone formation 100% of the time, that pluripotent stem cell derived osteoblasts have a much lower success rate. So this was an important lesson that we could not just rely on in vitro assays to predict osteoblast function and bone formation. Of course, the challenge in the bone field is that unlike hematopoietic stem cells where you can ablate temporarily with lethal irradiation and then transplant back a limiting number of stem cells to repopulate the entire hematopoietic system, you really don't have that option with the skeletal system. It's not possible to ablate the entire skeleton and then try to transplant that back. So we took the approach of something called organ complementation. The idea here is that mice lacking RUNX2 cannot form mature osteoblasts, and therefore in a skeletal preparation, where red is highlighting osteoblasts and blue is highlighting cartilage, RUNX2 null mice cannot form fully mature osteoblasts or a mineralized skeleton. We reason that if we intercross 2 RUNX2 heterozygotes and harvested their blastocysts at embryonic day 3.5, and then injected, these are wild type and it says ES or iPS cells, so either embryonic stem cells or induced pluripotent stem cells that were labeled with a fluorescent marker. If we injected them into the blastocyst, we would generate chimeric embryos. And of the resulting embryos, both wild type and heterozygous animals, we would expect the entire animal to be chimeric, including the skeleton. However, in those that were derived from RUNX2 null background, again, the skeleton cannot form from the host background since RUNX2 is required for osteoblast maturation. And therefore, any bone elements in osteoblasts we reasoned would have to be derived from these wild type stem cells. And we reasoned that this would be an in vivo rigorous assessment for the ability of stem cells to become bone forming osteoblasts. These were technically challenging experiments, and it took many years to accumulate even a few number of targeted embryos. But here I show in our hands, this is a wild type embryo at embryonic day 18.5, and you can see a beautifully mineralized skeleton. The RUNX2 knockout has no mineralized skeleton. We do see a little bit of mineralization at the site of hypertrophic chondrocytes in the knockout mice. And then we were very gratified to see that when we injected, these are in this figure, embryonic stem cells, that we had some reconstitution of several skeletal elements. You can see especially the mandible, parts of the limbs, and parts of the skeleton, vertebral column. Because these stem cells are labeled with YFP, we could take their soft tissues like liver and use flow cytometry to quantitate the percent contribution from these stem cells. And you could see that it matched the expression as visualized under fluorescence imaging in the skin. And here just showing the skeletal preparations of the skull, I think you can see that there's very little mineralized bone in mice with minimal stem cell contribution, but with increasing percent chimerism, we get more and more normal appearing skull. We also went on to show that these pluripotent stem cells can rescue the bone marrow hematopoietic niche, which is another area of interest in our lab. RUNX2 null mice do not form a marrow cavity, but when we introduced wild type pluripotent stem cells, you can see that the percent chimerism as viewed by GFP expression is at least 50%. And although not normal, there is some restoration of a bone marrow cavity with hematopoietic elements. And we were able to show for both embryonic stem cells and iPSCs that the greater the contribution from stem cells, the more normal appearing the bone marrow cavity. And we also used another approach where we crossed Ostrix Cree expressing mice to DTA, diphtheria toxin expressing mice. In this way, we could ablate all of the host derived osteoprogenitors as soon as they express Ostrix. Again, we introduced wild type pluripotent stem cells. In this case, the DTA is fused to GFP, so we used non-GFP expressing wild type stem cells. Again, in any chimeric animals in which Cree and diphtheria toxin are expressed, the skeleton should only be derived from the introduced pluripotent stem cells. As a control, you can see that in control animals, there are relatively normal limbs, but the expression of Cree and induction of DTA ablates normal skeletogenesis. And again, we were able to show that the contribution of pluripotent stem cells, as it increases, we get a relatively more normal skeleton formation. And the threshold appears to be somewhere between 40 to 50% contribution from these stem cells. So this demonstrates in principle that pluripotent stem cells can become, in fact, bone-forming osteoblasts in vivo. However, there are some reasons that you might not want to transplant pluripotent stem cells themselves, and perhaps the greatest is that they can form teratomas in vivo. And we showed this with our stem cells. They express markers of pluripotency like OCT4, and they spontaneously differentiate into all three layers of all three germ cell layers as shown by expression of their markers. And finally, if these cells are implanted into mice, they do form teratomas that again, histologically have elements of all three germ layers. So as our next approach, we thought maybe we can drive pluripotent stem cells to differentiate into mature osteoblasts in vitro first. And to do this, we relied on fluorescent markers generated by Andy McMahon's lab or David Rowe, in which various markers drive the expression of green fluorescent protein. And we harvested both ES cells and derived induced pluripotent stem cells from each of these lines. And the hope was that once we had differentiated them, we could use the GFP as an initial marker of osteoblast differentiation. So we have largely relied on David Rowe's collagen 2.3 GFP mice. In our hands, this is a very reliable marker for osteoblast maturation. Its expression increases with differentiation. And so we had an in vitro protocol for differentiating stem cells using embryoid bodies with retinoic acid. Differentiating them into osteoblasts, we were able to show that these cells in vitro can indeed form mineralized nodules, and that when we harvest these cells from col 2.3 GFP expressing mice, that these nodules express GFP under fluorescent imaging, and that we can use flow cytometry to detect GFP expressing presumed osteoblasts. However, as you can see, the yield is very low, less than 1%. We were able to harvest enough cells from flow cytometry to perform transcriptome analysis, starting with parental IPS and ES lines, comparing to calvarial osteoblasts, and then looking at both iPSC and ESC-induced osteoblasts. And we were gratified to find that these two pluripotent stem cell-derived osteoblast lines cluster more closely to calvarial cells than they do their parental lines. And that's visible here as well on the heat map. But this is not a feasible mechanism without a lot more optimization to get a large number of osteoblasts. So in the last few minutes, I want to talk about our approach using direct reprogramming. This is an approach in which induced pluripotency takes a differentiated stem cell, for instance, a fibroblast or a skin cell, pushes it all the way back up into a pluripotent stem cell state, and then you can differentiate from there into a different lineage. However, in direct reprogramming, many groups in different organs have published that one differentiated cell can often be induced to sort of hop over the wall into an adjacent or similar differentiated cell type. And this has been published for many different kinds of cells, including fibroblasts being converted into myoblasts by MyoD, myeloblasts being converted into other hematopoietic lineages, and B lymphocytes being converted into macrophages. So we reasoned that we could harvest fibroblast tail tip from the tail tips of collagen 2.3 GFP mice, that we could screen using lentiviral infection with a variety of osteogenic transcription factors, and screen for what we would call induced osteoblasts that express collagen 2.3 GFP. And a priori, we set several criteria that would have to be met in order to call these induced osteoblasts. They would, of course, express collagen 2.3 GFP because that's the screening assay that we use. They should express markers of osteoblast differentiation. You would expect them to form mineralized nodules in culture that are GFP positive. They should have a transcriptome and an epigenome that resembles those of osteoblasts. And most importantly, they should form bone in vivo. And so we were able to screen through a large number of transcription factors. We identified a set of 4, RUNX2, DLX5, Ostrix, and ATF4, that when introduced into tail tip fibroblasts, robustly induced GFP expression. And you can see here that the col 2.3 GFP expression rises significantly in these transfected cells with differentiation. We could also show that the expression of osteoblast markers like col1A1 or osteocalcin also increases with differentiation to approach that of calvarial osteoblasts as a positive control. They form mineralized nodules robustly in culture, shown here stained with alizarin red or von Casa compared to mock infected cells. And under immunofluorescence, you can see that the col 2.3 GFP co-localizes with expression of osteocalcin. The transcriptomes do indeed cluster more closely to the calvarial osteoblasts than to the parental fibroblast line. And we showed similarly that the epigenetic changes also resembled those of calvarial osteoblasts. And finally, if we take these cells and implant them with matrigel in subcutaneous nude mice, over several weeks, they can form bony elements only with the induced osteoblasts and not with or very minimal from the implanted tail fibroblast controls. And this is micro CT imaging to show some bony formation. And then under histology, you can see that they stain for alizarin and Boncasa, and that these nodules do indeed express GFP in nodules that are also expressing osteocalcin. So in summary, I've shared with you today that we have been interested in the role of PTH receptor signaling in the lineage allocation of MSCs into osteoblasts versus adipocytes. We've also been very interested in whether we can drive the differentiation of pluripotent stem cells into osteoblasts. And the answer is yes, but currently the yield is low and a great deal of optimization would be needed to make this, I think, a clinically applicable strategy. However, we've had a greater success with direct reprogramming of osteoblasts. This appears to be a much more efficient source of osteoblasts. Looking ahead, we're excited for the potential regenerative medicine applications, thinking about approaches using 3D printing with biomaterials for tissue engineering. And with that, I want to thank the members of my lab who have done the work, particularly the pluripotent stem cell differentiation and the direct reprogramming work has all been done by Hui Zhu, a very talented staff scientist in my group. And I want to thank my sources of funding for supporting our work over these many years. And with that, I thank you all for your attention, and I'm happy to take questions.
Thanks so much, Joy, for your great presentation. We have about 10 minutes for our question and answer session, so feel free to unmute yourself or submit your question in the chat. Thank you.
And I apologize if you hear the construction behind me, but of course they chose today to renovate the lab next door.
Hello. Nice presentation. So your conclusions were that the PTH1 receptor would help kind of steer the stem cells, mesenchymal stem cells, into the osteoblast lineage, which would then, if there was a deletion in that receptor or the subunit, lead to severe osteoporosis. Can you make the distinction between, say, what happens at the embryonic stage and what would happen with hyperparathyroidism? Are there similarities between the two?
Yeah, that's a great question, Rhonda. So we have in our mouse models largely been looking at constitutive deletion through embryonic. So it's definitely a developmental model. That being said, these mice are tetracycline or doxycycline regulatable. So we can defer the deletion of either PTH receptor or GS until the postnatal stage. And we have been able to generate some adult mice lacking PTH receptor signaling in that. And we have shown that they have pretty severe osteoporosis. We've shown that they can't respond to anabolic PTH in terms of increasing trabecular bone mass if they don't have PTH receptor signaling. We have not investigated the adipocyte phenotype in as much detail as we did for the embryonic model. But in some sort of preliminary studies, it does look like they do have a pretty dramatic increase in adipocytes as well. And that would fit, I think, with what we know clinically, which is that osteoporosis induced either by aging or other conditions like glucocorticoids can be associated with an accumulation of marrow adipocytes. Specifically for primary hyperpara, that's an interesting question. And you would, in a way, that's now the model of continuous sort of exposure to PTH, and they have a bone loss due to resorption. I'm not familiar, and I don't know if anybody else knows whether the marrow adipocyte sort of profile has been carefully studied in these patients, but I think it would be a very interesting question.
But do you think in terms of the subunit, in your deletion model, it was the absence of the G-stimulatory subunit that caused osteoporosis or led to osteoporosis? Do you think in hyperparathyroidism, is there some type of maybe overstimulation of that subunit that causes it to be dysfunctional ultimately? That would
Yes.
Osteoporosis?
So I think that'll be an interesting thing. It's complicated by the fact that, of course, the kinetics of PTH exposure influence the outcomes, right? So in our hands, loss of PTH receptor, we've done, for instance, see a few assays to show that mesenchymal stem cells appear to be biased towards the adipocyte lineage rather than osteoblast lineage, which fits with the phenotype. In overexpression of PTH, like hyperpara, some of that bone loss is driven by increased resorption in osteoclasts. And so I think we would have to purify out the mesenchymal stem cells to look specifically at what's going on in that lineage.
Okay, all right, thank you.
Hello, Dr. Wu. You showed that very convincingly that your directly reprogrammed cells make bone in vivo. Was that bone in subcutaneous areas, and if you were to inject it in, say, a model of fractured bone, would they localize? So I guess where I'm going with the question is, are biomaterials necessary for something like healing a bone union, or would the cells remember enough that they are osteoblasts to kind of form organic structures themselves?
Right, that's a great question. So this was an ectopic in vivo bone formation assay. So we basically mixed the cells with Matrigel and injected them under the skin. And over a few weeks to months, they will form bony nodules. And it was basically a quick and dirty way to see if they could form bone at all. I think a much more elegant approach would be what you're proposing, which is, for instance, fracture non-union or a critical defect. In other words, in mice, you can induce a critical defect either in the calvaria or in the long bones, in which you induce a defect that's just basically too large for endogenous osteoblasts to fill in. I would need some help from some of our bioengineering colleagues about the right materials and the right approach. But I think my understanding is that in those circumstances, the purpose of the materials is sort of to hold the cells physically in place to bridge that gap until they can differentiate and restore the bone.
Can I jump in and ask
Of course.
a question? So of course, it's very interesting talk. I liked it very much.
Thank you.
My question is something naive. I want to ask you is you did this inducible osteoblast formation. So in this method, do you see any other cells coming up, like apart from osteoblasts and fibroblasts? Do you see any other mesenchymal cells?
So it's a great question. And whenever you do pluripotent stem cell differentiation, I think it's very messy, right? What's coming through from that process is, of course, we only had about 1.5% osteoblasts. So who knows what's in the other 98% of those cells. It is the reason we have relied so heavily upon those 2.3 Col1 GFP reporters, because they have been, and in other groups that have used direct reprogramming, for instance, here at Stanford, we have a group that has worked on reprogramming into neurons. They've relied on a similar approach to use a GFP marker. So I think that our goal was just to see whether we could get to the final osteoblast stage. But I would not be at all surprised if other mesenchymal lineages were in there. And I think depending on how we might optimize the differentiation protocol, it sounds like you're getting at, could we actually leverage that, right? Could we maybe use that to drive other lineages or something like that? And I think that's a very interesting area of study.
Do we have any more questions for Dr. Wu before we move on to the next presentation? All right, well, Dr. Wu, once again, thank you so much for a great presentation and a great Q&A session. I'd now like to turn over to Dr. Ramasamy to present his research.
Thank you. I would like to thank ASBMR for this opportunity. It's fantastic to connect with you all through this meeting. Let me share my slides, please. Yeah. Okay. Could any one of you confirm that you can see my slides?
Your slides look great, Dr. Ramasamy.
Great, thank you. So I'm Saravana Ramasamy. So I'm going to talk to you about blood vessels in the skeletal system today. So fundamentally, my lab is interested in understanding blood vessels. So you probably all know blood vessels are the structures that supply oxygen and nutrients to various organs and tissues in our body. So apart from that, blood vessels also perform a lot of tissue-specific and organ-specific functions. So what I mean by that is here I have 4 organs and their blood vessel images here. So you can appreciate that in lung, the blood vessels are involved in the exchange of gas, and in kidney they are involved in exchange of fluids. Okay, so that's kind of tissue specific functions they do apart from supplying nutrients and oxygen. So not only the organ-specific functions, so in pathological conditions, the blood vessels in organs are modified. And if you take any injury repairs situation, the blood vessels are the structures which are always modified. So we know that bone performs a number of functions. So specifically, we know that, so I have listed few physical and physiological functions on both these sides. I don't have to explain all this to this community, but we don't know how the blood vessel functions or how much they contribute to these physical and physiological functions of bone. So that's basically the aim of my team, to understand functions of blood vessels in bone health, disease, and repair. So how we do that? So we start from understanding blood vessels and how they interact with bone. So we know that the blood vessels are basically composed of endothelial cells and perivascular cells. So endothelial, we study how endothelial cells interact with other cells in the bone, and in turn, like how the other mesenchymal and hematopoietic cells in the bone interact with the blood vessels. So we mainly use mouse model system because mouse is very easy to manipulate cell biology because of availability of a lot of genetic mouse models. But of course, we are very much interested in understanding or studying clinical samples. So if you are interested in my research or science, please feel free to contact me. So now I just jump into explaining the organization of blood vessels in bone. So if we just cut open mouse bone and look at the blood vessel organization. So here my image is, the green structures are our blood vessels. So this is taken using a transgenic mouse model, which is combining weak gathering Cre-RT2 with reported GFP. So you can see the blood vessels in bone are highly heterogeneous. And if you look keenly, you can see the blood vessels just below the growth plate are kind of linear structures. And in the endosteum region, also they are linear structure, while here in the center of the bone, in the diaphyses, the blood vessels are highly branched network. So I should say that although they have different structures, they form a single vascular network. So what I mean by single vascular network is that the blood flows through them continuously. So they are not forming a different circulatory system. They are basically single vascular system. So if we then we use different markers to characterize them. So finally, we found that CD31 and endomycin are very good markers to separate these two structurally different capillary subtypes in bone. So CD31 is actually a panendothelial cell marker, but in bone they mark all the blood vessels, but also mark a lot of hematopoietic cells. So here